Hi everyone, my name is Shellen McCoy. I just want to say welcome. And on behalf of the Foundation for Conservation of Atlantic Salmon, I just want to say welcome to today's seminar. So we always say thanks to the Government of Canada because of them we're able to do this with their contribution. And just to go right into our session today, I just want to say welcome to Dr. Tara Himlay. Welcome to be here with us. Dr. Imlay has a PhD from Dalhousie University. She has a master's from Acadia University and a bachelor's from University of Guelph. Her past research has largely focused on understanding the drivers of population change for species and evaluating the efficacy of conservation actions on species recovery. This work spans several taxa, including insects, reptiles, birds, and fish in Canada and the USA and Mauritius. Today, she's going to be speaking on spatial synchrony and body length changes for Atlantic salmon, Salmo Salar, over 5 decades. After this afternoon's presentation, of course, we'll do like we always do. We'll open up for questions and answer, and I'll help monitor and manage that part of the session. But with no further ado, I'm going to pass it on to Tara to do her presentation. So welcome, Tara.
Thank you. I'm really excited to be able to share the work with everybody who is able to attend today. However, before I get started, I just want to take a moment to acknowledge that while this seminar is hosted in a virtual platform, I want to acknowledge the Indigenous peoples of all the lands that we are on. I live, work, and play on the lands of the Tsawwassen First Nation, as well as on the traditional unceded territories of the Musqueam and other Coast Salish peoples. I encourage you to take a moment to acknowledge the Indigenous peoples of the lands where you live, work, and play. We do this to reaffirm our commitment and responsibility to improve relationships between nations and our own understanding of local Indigenous peoples and their cultures. The work that I'm presenting today involves a large collaboration of people throughout Eastern Canada. They've all contributed data from long-term monitoring programs by provincial and federal government agencies, and I've listed them here on this slide. Because this work spans salmon populations throughout Eastern Canada, it would not have been possible without these contributions. Additionally, this work is also supported by a DFO CSRF grant. So you might be wondering why we're interested in body size. There's often a hyperallometric relationship between body size and reproductive output. And reproductive output can include factors like the number of eggs, egg volume, and energy in eggs. This hyperallometric relationship means that at larger body sizes, individuals have a much higher reproductive output than what is proportional to their body size, as you can see from the blue line here on this figure. This figure is an example of these relationships for Atlantic cod. So under the isometric scenario, larger cod still have higher reproductive output than smaller individuals. However, under that hyperallometric relationship on the red line, you can see that reproductive output increases more rapidly with increasing size. The result of this hyperallometric relationship is that the reproductive output of a single 30-kilogram cod is equivalent to 37 2-kilogram individuals. So this is more than double the mass. And this relationship forms the modern fisheries management, and larger individuals are recognized as being disproportionately important to population management than smaller individuals. Further, this relationship is often a key consideration in establishing biological reference points for tracking the stability of stocks. So over the next couple slides, I'll provide examples on some of the factors that can result in changes in body size. The first of which is temperatures. So warming temperatures, particularly those associated with climate change, are predicted to result in faster growth, but earlier maturity for a variety of taxa. And ultimately, this will result in smaller body size. One study estimated that for each 1 degree Celsius increase in temperature, there would be a corresponding 6 to 22% decrease in fish body size. So the figure here on this slide shows the predicted change in body size globally, with warmer colors indicating areas where body size decreases are anticipated. And when this is considered across the latitudinal gradient on the right panel, these body size declines are predicted to occur across most of that latitudinal gradient. Another factor that can result in changes in body size is density dependence. This density-dependent relationships can affect body size through increased inter- or intraspecific competition for resources such as food. And it's estimated that 54% of marine fish stocks experience density dependence during the late growth period. One example of this is for Chinook salmon populations in Alaska. And if you look at the left panel, the authors of this study found that the body size of Chinook salmon decreased as the abundance of Chinook salmon increased. Also, as the abundance of another closely related species, the pink salmon shown in the right panel, increased, the body size of Chinook salmon also decreased. A few other factors can also result in changes in body size, and these include the abundance or fragmentation of available habitat, food availability, and any size-selective mortality or harvest that's directed towards either the smallest or largest individuals. Synchronous changes in body size can arise when you have a synchronizing factor that either acts across a sufficiently broad scale to cause synchronous changes in body size for multiple populations, or when you have synchronizing factors that operate on multiple populations when those populations are concentrated in space and time. And we'll return to this when we start talking a little bit more about Atlantic salmon. But first, I'm going to present a really simplified life cycle of Atlantic salmon that just focuses on the two most common adult age classes. So salmon spawn in freshwater rivers throughout North America and Europe. And after 1 to 8 years developing in freshwater, they undergo physical, physiological, and behavioral changes that allow them to survive in the ocean. Upon entering the marine environment in the spring and summer, they spend 1 or 2 years in the ocean before returning to freshwater to spawn. And so individuals that spend 1 winter at sea, I'll refer to as 1 sea winter individuals, and those that spend 2 years are 2 sea winter individuals. For salmon, the fastest growth rates and largest increases in body size occur while they're feeding in the ocean before they return to spawn. And unsurprisingly, 2 sea winter individuals do attain larger body sizes than 1 sea individuals because they spend more time in the ocean. And the difference in mean length is about 210 millimeters between 1 sea winter and 2 sea winter individuals. So upon entering the marine environment, salmon from North America travel towards shared feeding areas east of Newfoundland and in the Labrador Sea. Following spring, a large proportion of this population will mature and migrate back to rivers to spawn, and these are the 1 sea winter salmon. The remaining salmon will migrate north in the Labrador Sea during the summer and are known to congregate around Greenland. Additionally, some individuals will also migrate to the eastern North Atlantic Ocean and are known to congregate around the Faroes. Most of these individuals then return to the rivers to spawn after their second winter. So the figure on this slide shows the abundance of North American salmon since the 1970s, which is when most monitoring programs were initiated. We know that similar declines are also occurring in European populations. It's thought that these declines are likely due to changes in the marine environment. And past studies that have examined relationships between potential factors in the marine environment and population trends or demographic processes have actually noted a high degree of synchrony or common changes across populations, including synchrony in population trends, productivity, ratios of different aged individuals, and other factors like survival, although there's some mixed support for that, and marine growth rates. So I'm going to share some findings of a recent study on marine growth rates for salmon in Norway. This is the most geographically extensive study on growth rates of salmon, and it covered a gradient of rivers that spanned about 11 degrees of latitude. The authors of the study divided that gradient up into 4 different regions, each indicated by the different colored ellipses on this figure. The researchers found that there was actually a sudden decline in the growth, particularly for the 2 southern regions, those in blue and purple, between 2004 and 2005. And the authors of this work linked these changes to the reduced volume of Arctic water entering the Norwegian Sea, warmer ocean temperatures, and the decreased availability of zooplankton and growth of mackerel in the area. So we were interested in looking at whether or not there have been synchronous changes in the fork length of Atlantic salmon throughout eastern Canada, and whether or not any changes in fork length were related to broad-scale marine factors, including those that could be driving synchrony in these populations. We did focus on spatial synchrony for 1 sea winter and 2 sea winter individuals, just from factors that are occurring in the marine environment, because this is where changes, the largest changes in size, are occurring. So over the next couple slides, I'm going to focus a little bit on the data that goes into our model. I'm going to start with some of the biological data on salmon body size, and then I'm going to talk a little bit about the environmental covariates that we considered that could drive synchrony. So we obtained biological data on Atlantic salmon from 46 rivers throughout eastern Canada. And this the periods of sampling varied a little bit by river, but we did have data for at least some period of time from 1970 to 2022 for these rivers. The plot on this slide shows the locations of those 46 rivers that we included in the database, but only the named rivers were included in the analysis after we removed individuals that lacked data on size or age, individuals from hatchery, aquaculture, captive breeding, and smolt to adult supplementation programs. And we removed data from any year where fewer than 10 1 sea winter individuals or 10 2 sea winter individuals were sampled. And then lastly, we only included the rivers that had a minimum of 20 years of data. So after filtering this data, we were left with still over 2,000 1 sea winter and 2 sea winter salmon from 19 rivers throughout eastern Canada with a minimum of 20 years of data. So the plot here just shows the available data from each river from 1971 to 2021. And these rivers are arranged in order of increasing latitude. So the LaHave River at the bottom of this plot is the most southern river, and the English River is the most northern river. We included all 19 rivers in our analysis of fork length change for 1 sea winter salmon, but we only had sufficient data from 8 rivers to be included in the 2 sea winter analysis. And all of these rivers are indicated by asterisks beside their names. If you're familiar with these areas, you may notice that none of the rivers from Newfoundland were included in the 2 sea winter analysis. And this is largely due to the fact that returning individuals to these rivers are primarily 1 sea winter individuals. So we just didn't have sufficient information on the 2 sea winter individuals in these populations. So for each river and year, we then calculated the mean fork length, and these mean values were included in the analysis. So next we considered a couple different mechanisms for body size changes, and we focused on covariates that could drive synchrony in body size, which included both broad scale environmental conditions and conditions at some of those shared locations. We wanted to recognize that there's differences in the timing and space use of individuals during both their first and second years at sea. And so we incorporated that into our covariates as well too, and you'll see that in a greater detail in the next couple slides. But ultimately we were left with 5 different mechanisms for body size change that we explored in greater detail, including climate, thermal habitat, food availability, density dependence, and fisheries exploitation rates. So I'm just going to walk you first through the plot on this slide, which is going to be similar to some of the plots that you'll see on the next couple slides. So this plot just shows the values for the 3 different climate indices that we considered in our analysis for salmon during both their first year and second year at sea. The values for each index have been centered and scaled so that they have a mean of 0 and a standard deviation of 1. The first climate index that we considered was the Atlantic multi-decadal oscillation. And this is a measure of sea surface temperatures anomalies, and it generally varies over multi-decadal scales. You can see during our time series, there was a switch around 1995 when AMO changed from a relatively cool phase to a warm phase. The next climate index that we considered was the North Atlantic oscillation, which measures pressure differences between Iceland and the Azores. Positive phases of NAO generally represent cooler temperatures and less precipitation in Canada, Greenland, and Southern Europe. And then lastly, we considered the Newfoundland and Labrador Climate Index, which is a relatively new index that's available for measuring climate conditions in the Labrador Sea and sort of the Western North Atlantic Ocean. Positive values of the NLCI represent periods with warmer air and ocean temperatures, more precipitation, less sea ice, and fewer icebergs. So next, we looked at thermal habitat to represent the growth potential of ocean temperatures. And we weighted sea surface temperatures to reflect the non-linear relationship between temperature and growth. So during both the first and second year at sea, when sea surface temperatures were less than 1 degree Celsius, we assumed that there was little growth potential, so this was set as 0. Next, between 1 to 6 degrees Celsius, there was a linear increase in the growth potential of the habitat. And then peak growth from 6 to 14 degrees Celsius during the first year and 6 to 12 degrees Celsius during the second year. Then there was a linear decrease in growth potential from 14 to 18 degrees Celsius during the first year and 12 to 15 degrees Celsius in the second year. And then lastly, temperatures above this threshold were assigned a value of 0. So next, using information on the movements and distribution of salmon in the North Atlantic Ocean, we identified the marine range of 1 sea winter and 2 sea winter individuals. So each dot on this map here represents a 1 by 1 degree block in that area. So the yellow half circles indicate the marine range of 1 sea winter individuals during that first year. And then the 2 sea winter individuals, or the second year, you can see the expanded range that extends further north into the Labrador Sea and eastern to the Faroes. And that just helps to encompass the transoceanic migrations that we know some of these individuals undertake. And then lastly, we calculated thermal habitat over 4 periods during both the summer and winter for both years. And so these plots here show those time series values for our 4 indices of thermal habitat. And you can see that since 1971, summer and winter thermal habitat in the first year, so those are the 2 leftmost panels, and then winter thermal habitat in the second year have all experienced increasing trends. Next, we looked at food availability. So salmon forage across a really broad prey field that changes both through the year and as individuals increase in size. Generally, they forage on larger individuals as they grow. So for our analysis, we included zooplankton as a measure of both secondary productivity and first year diet using information from the Continuous Plankton Recorder Survey. And then we included capelin as a measure of both first and second year diet. Both of the data sets that we used here did have some missing values for some years. And so we either address these missing values using either a general additive model in the case of the zooplankton, or a principal components analysis to interpolate missing values for capelin. And I won't get into those details today, but if anybody has any questions on them, I'm happy to explain that further. Then for our complete time series, we then used a principal components analysis on the zooplankton and the capelin information to derive a single index of food availability. And in this case, we used the first principal component from these two principal components analyses. You can see that a really high proportion of the variability in the data was encompassed by this first principal components analysis. So the first PC for the 1C winter encompassed almost 85% of the variability. And for the 2C winter individuals, it was nearly 79%. The trend in this principal component is also provided here on this slide. And what this represents is that periods of the relatively high index represent years where the biomass of zooplankton and capelin was higher. Next, we looked at density dependence, and we used the reconstructed marine abundance data from the 2023 ICES report. Density dependence generally arises among individuals of the same size or age. So we looked at two different indices of density dependence for each year at sea. In the first year, we just included all North American 1C winter salmon as our measure of marine abundance. And then in the second year, to encompass those transoceanic movements of salmon from both continents, we included the marine abundance of both North American and Southern European non-maturing 1C winter salmon. So these are individuals that remained in the ocean for the second year. They didn't return to rivers. And probably unsurprisingly, given that we know that salmon are declining across the marine range, we do see that there's a decreasing trend for marine abundance in both of these indices. And then lastly, we obtained catch data from mixed stock marine fisheries, specifically those that are known to harvest salmon from North American populations throughout our latitudinal gradient. These are also all size selective fisheries that use gill nets and are known to selectively harvest larger individuals. So the first group of fisheries that we considered were located around Newfoundland, Labrador, St. Pierre, and Miquelon. And these fisheries largely target individuals on their return migrations to rivers. So for the first year, we looked at the exploitation rates of small salmon, and in the second year, we looked at the exploitation rates of large salmon. And then you can see on these that there's a sudden drop in exploitation rates within these NAC fisheries. And this is due to fishery closures that went into effect in Newfoundland in 1992 and Labrador in 1998. So present day catches in this region are primarily from Indigenous food, social and ceremonial fisheries, and subsistence food fisheries for Labrador residents. And then the commercial and recreational fisheries in the territorial waters of St. Pierre, Miquelon. The second group of fisheries we looked at were around Western Greenland. And these fisheries primarily harvest individuals that do not mature after the first year. So these are only individuals that are present during the second year. So we didn't calculate exploitation rates for this fishery for the first year. Similarly to the other fishery, there was a sudden drop in exploitation rates for the West Greenland. And this is due to an export ban that went into place at 1998, after which this fishery only harvests salmon for consumption within Greenland. So just to summarize all those potential covariates that we're considering, so for 1C winter salmon, we just focused on the covariates that affect them during the marine period in the first year. And then for the 2C winter individuals, we included covariates for both the first and the second year. You can see that most of these covariates are the same for 1C winter individuals in the first and second year, with just a few differences. including food availability, because we integrated our food availability index across both years for the 2C winter individuals, so there were some differences there. For density dependence, we have an additional component of density dependence for the second year. And then for fisheries, there were some differences in those covariates as well too. So using the information on body size and these environmental covariates, we used a dynamic factor analysis to model trends in fork length through time. And DFA models can be divided into 3 different components. The first component looks at common trends in the data and provides both common trends, 1 or more, as well as loadings for each river that was included in the analysis to represent the strength of that common trend. We can also incorporate covariates into DFA models to explain changes in fork length over time in a similar manner as to other linear models. And then the third component is the observation error, and I won't spend much time discussing that part today. When we were considering how to best incorporate the covariates we're considering into our models, we actually found that there were quite strong correlations between many of these covariates. And this was not particularly surprising because there have been several previous studies that have published many of these relationships. However, because of these strong correlations, we chose to individually consider relationships between fork length in each of these covariates rather than trying to combine multiple covariates into a single model. So we did our DFA in 2 different steps. Our first step, we just focused on identifying common trends, the number of common trends that best fit the data, and the different variance, covariance structures to address the observation error. Once we had developed that best model for explaining changes in fork length based on a combination of common trends, we then incorporated additional covariates into the model in a sequential manner and identified any covariates that improved on that base model that we identified in our first step. So before I get to the results, I just want to provide a brief explanation of what these loadings and common trends look like. And for an example, I'm going to use the results for fork length of 1C winter salmon from the Miramichi River. So our best DFA model for 1C winter salmon included 3 different common trends, and that's the figure that you see on the left side of this slide. Those are each of those different common trends. And then for the Miramichi, there is a loading for each of those different trends. And you can see that the loadings for the first and third common trend were much higher than the second common trend, indicating that the overall trend for the Miramichi River is largely going to come from a combination of the common trends number 1 and 3. And so each of those loadings is multiplied by its corresponding trend, and then those values are summed to obtain the overall trend for that time series, which is what you can see here on this plot. So this is all the results for the common trends of 1C winter salmon. And I'm going to take a bit of time now to walk you through this plot. So on the x-axis, I have the rivers arranged by latitude, starting with the LaHave River at our lowest latitude, the English River at our highest latitude. The first row of plots are those common trends. The first common trend, the second one, and the third one. And while there are differences between these common trends, all 3 of them do actually suggest that there were increases in fork length that occurred during the 1980s and 1990s. And then for the first and second common trend, we do see some evidence of a declining trend through time, although this is relatively stable for the third common trend. The second row of plots here shows the loadings for each trend and river. And if you look across all of these, each river generally had a relatively high loading for 2 or 3 trends, suggesting that the overall trend for that river is a combination of these common trends. But if we focus solely on the highest loading for each river, so that's the blue bars that you see here, we do see a little bit of separation by latitude. So for the first trend, the loading was often highest for the most southern rivers, particularly those in the Bay of Fundy and around the southern coast of Nova Scotia. The second trend is often highest for rivers in the southern Gulf of St. Lawrence and southern Newfoundland. And then lastly, the third trend was often the highest for northern Newfoundland rivers. And you may have noticed that a few of these loadings are negative. And so when loadings are negative, this just indicates that the river has the inverse of the common trend that you see. So in the next 3 slides, I'll show the resulting trends for each river. So these are the rivers that had their highest loading for the first common trend. And so you can see in the left-hand side of this plot, I'm showing the first common trend, as well as the loadings. So these are the rivers that have the blue bars here. The blue points in the panel on the right-hand side of this plot are the mean values for each year. And the size of those points just indicates the number of individuals that were included in that mean. For these 6 rivers, only the Miramichi had a significant increase in fork length of about 55 millimeters during the 1980s and the 1990s. So these are the rivers that had their highest loading for the second common trend. And there was a significant increase in fork length for salmon in the Trinity and Haut Rocher rivers during the 1980s and 1990s, and a significant decrease in fork length for salmon in the Trinity after about 2000. And then these are the trends for rivers that had their highest loading for the third common trend. And there was a significant increase in fork length for salmon in Middlebrook, Gander, and Torrent rivers during the 1980s and the 1990s. Our DFA for the 2C winter salmon resulted in a best model that just had a single common trend. So like we saw for the 1C winter individuals, there was also some evidence for different trends across the latitudinal gradient. And in this case, 3 of the 4 rivers that discharge into the southern Gulf of St. Lawrence, so the Miramichi, St. John, and Trinité, had higher loadings than the more southern and more northern rivers. However, only the 2C winter salmon from the Miramichi River had a significant increase of about 48 millimeters during the 1980s and 1990s. So now that we've identified our base model, we then started to look at the covariates that could improve the fit of this model. So for 1C winter individuals, only the inclusion of exploitation rate resulted in an improvement to our base model. And we found that there was a significant negative relationship for 9 of the 19 rivers. And this just indicates that when exploitation rates were lower, salmon had a longer fork length. For 2C winter individuals, we actually identified 6 covariates that improved the fit of the base model for 2C winter salmon. And I've arranged these covariates in order of the greatest improvement to the model fit, which was thermal habitat in the second winter, to the North Atlantic oscillation, which had the least improvement in model fit. Collectively, the relationships between fork length and thermal habitat and most of the climate indices, AMO, NLCI, and NAO, suggested that when the habitat had a higher growth potential or when conditions were warmer, salmon were longer. Although you'll see exploitation rate, when it was included, it did improve the model fit. There actually was not a significant relationship between fork length and exploitation rates for any of the rivers. So the 2 key findings that come out of these results are that there was a relatively weak spatial synchrony across the latitudinal gradient, but we did see a little bit of separation between our southern, mid-latitude, and northern rivers. We also saw that there's an indication that fork length did increase during the 1980s and 1990s, and for some rivers has started to decline after about 2000. This weak spatial synchrony was a bit of a surprising result, given that there's been so much documented synchrony in population declines, life history traits, and other demographic processes and other analyses. And we considered a couple different reasons for why this might be occurring. The first is that there could be river-specific factors that have a much greater effect on body size changes than the covariates that we were considering. Similarly, differences in the timing of marine entry or conditions encountered during migration could also have a greater effect on body size changes than the covariates we considered. And these two factors would result in weakening spatial synchrony. And then lastly, we know that individuals during the second year use a very broad region across the North Atlantic. So if different populations are exploiting different areas during this time, this could also weaken spatial synchrony as well too. However, we did find relationships between thermal habitat and climate indices during both the first and second years at sea and the length of 2C winter salmon. These factors are largely related to metabolism, and most of these relationships suggested that warmer temperatures were related to increased body size. This is contrary to findings from European salmon populations and predictions in the literature around the effects of warming temperatures on salmon populations. And while we don't know exactly why this is occurring, we do speculate that temperatures are generally cooler in the Northwest Atlantic compared to the Northeast due to several cold water currents that enter this area. So it's possible that temperature increases in this area haven't reached a threshold that could result in decreased body size. Alternatively, growth is generally more limited by food than temperature, and salmon forage on a really broad prey field. We weren't able to encompass that full prey field into our index of food availability. So it's possible that some of these relationships are driven by some measured component of food availability. We did not find any relationships between marine abundance and changes in body size. However, we did see that higher exploitation rates from the gillnet fisheries around Newfoundland, Labrador, and St. Pierre-Miquelon were related to lower body size. The fishing moratoriums that have occurred in Atlantic Canada during the 1990s likely resulted in these increases in body size that we observed during this period. However, it is important to note that exploitation rates are presently at historic lows and have been since the 1990s. So they're unlikely to be driving any present day declines in body size. Of course, our work had many limitations, including the fact that many of our covariates were strongly correlated with each other. So we were unable to disentangle direct and indirect effects of each covariate. Additionally, our data was focused on sampling individuals that returned to rivers, and so we don't have any data on individuals that died. So we don't know if there's any particular growth patterns or covariates that could lead to much reduced growth and lower survival. Additionally, because our DFA used mean values for fork length for each year, we couldn't account for any intra-year or inter-river variability. But we know that sampling for these rivers is relatively consistent across time, so we don't anticipate that sampling variation is driving the results. And then lastly, very few of the rivers that were included in our analysis were sampled across all 5 decades. So it's possible that we were unable to detect some changes in body size or relationships with certain covariates due to the periods of time when some of these rivers were sampled. So as I showed earlier in this talk, the relationship between body size and reproductive output is important for fisheries management. And Atlantic salmon populations have been in decline since the 1970s, despite the increases in body size that were observed during the 1980s and 1990s. This suggests that increases in size and the resulted changes in reproductive output were insufficient to offset losses due to other factors. Concerningly, we're starting to see evidence of declines in body size, some of which were significant for salmon stocks at mid-latitudes. But unlike what was observed in Norway, where there was that rapid change in marine growth between 2004 and 2005, the changes that we're observing in Eastern Canada are happening relatively slowly. So in the analysis that I've presented today, we looked at changes in the size of salmon for the two most common ages. However, I'm sure you guys are all aware, salmon have a really complex age structure and a diverse array of life history strategies. And this diversity is thought to be important for resilience to change for populations. Therefore, in our subsequent analysis, we'll actually be examining whether or not changes in size of the salmon of the population as a whole are occurring. And we'll also be looking at whether population changes in body size can be explained by changes in age structure and the life history diversity of the population as well too. And then ultimately we'll consider the implications of changing body size on the reproductive output of Atlantic salmon and the biological reference points that are used to monitor stocks. So I'm happy to take any questions or comments at this point. I've also included my email address on this slide. So if after the session today, if you have any questions about this work or you think of something that you wanted to ask me, please feel free to reach out.
Well, thanks, Tara. That was great. So it is time for questions and answers. We still have 10 minutes here that we can allow some. There's a couple of ways to do that. If you want to ask your own question, you can raise the little hand and I will unmute you and then you can say it out loud. Or if you want to type it in, I can read it out loud for you so Tara can answer. So we'll give folks a minute here and I'll check to see. And we do have a hand, so Tommy, just a second. Oh, there you go, Tommy, go ahead.
Hi, thank you. Thanks, Tara. That was a very, very good presentation. Really looking forward to this. A couple of questions. I am sure by way the graphs were constructed that this has been sent or perhaps even already published somewhere. I would really be curious to really clear presentation, but of course always miss a few details. This would most definitely be a paper I would be interested in reading, so perhaps you can inform where we will see this interesting paper coming up. And I'll ask my second question at the same time. Did I see that the Restigouche River was not included in the dataset? I'm a little bit surprised because that's one of the prime rivers where my understanding is that DFO and the Guardians of the River have been pretty much on page collecting data. So any comments on? So I can only assume that Restigouche actually then in the end did not have enough data.
Yes, unfortunately, we didn't have enough data to include the Restigouche in this analysis. It is in our database, but it just didn't meet some of those criteria for including it. We didn't have 20 years of size information for the Restigouche. So that's why it unfortunately got dropped.
Okay.
And in terms, this work has been submitted to Ecology and Evolution and it's presently in review. So fingers crossed it'll come out at some point.
Great, thank you.
Is there any other questions? Here. And I'm sure that Tara will welcome any questions. I know sometimes after you leave and the day of or the next day you thought, oh, I should have asked this. Please feel free to send it my way or write directly to Tara if she's okay with that. Her email is on the screen. Okay.
I have an additional comment, if I may.
Sure.
It is not a question, and I don't have an answer, but I'm just curious what your thoughts are on this. You mentioned that the current fisheries pressure is anticipated to be at very low level since the closure of the marine fisheries. Sure, there's the West Greenland component that you included in the 2C winter analysis. However, there is a paper, I'm sure you're aware of it. The lead author is Mike Dadswell, who and that group of authors strongly argued that there indeed is a fishery, what they consider to be illegal, unreported, and unregulated fishery, that is the culprit of all of this. Of course, when it's unreported and unregulated, there's no knowing if it's there and if these authors and their proposition is true or not. But should there be that fishery, could that explain some of the patterns that you see in your data and analysis?
Yeah, it's a great point. I have read that paper, the one that you're referring to. I think it came out in 2020, I think. And of course, the challenge is we don't really have good information, but for any fisheries to be driving changes in body size, there would have to be a size selective component to it. So that would have to be, either the equipment that's being used to selectively target larger salmon or possibly smaller salmon, but in this case, we're seeing more recent declines in body size for some populations. Yeah, so it would either need to be, some type of gear that's resulting in that size selective harvest, or it would have to be occurring in areas where larger salmon are congregating. So if we had more information on what was going on and those criteria, one of those criteria were being met, it's certainly possible, but I can't really speculate beyond that.
Because if I understood some of your data, again, like there's of course variability among rivers, but one of the conclusion bullets suggested that there seems to be this decline in the size since the 2000s or something like that. Mm-hmm. These authors do argue that fishery would be happening at the quote unquote high seas. Mm-hmm. That would kind of, it would not exclude the hypothesis that these authors are, because they predict that if there would be this unreported fishery, there should be a decline in size, which, if I understood it correctly, you're seeing in the data set.
Yes, but for some of the rivers, there's a more recent decline. Yeah, it was only significant though for the Trinité.
Yep. Okay. Thank you. Excellent presentation. I'm really nice to see this work.
I'm glad you enjoyed it. Thanks for coming.
Thanks, Tommy. We do have another question. I'm going to unmute Guillaume Dauphin. Guillaume?
Hello, can you hear me?
Yeah.
All right. Hey, everyone. Thank you, Tara. Tara works. I work with Tara on this project. Just on Tommy's last point, and and the Dadwell paper. So the Dadwell's paper is interesting as it gives a very interesting, hypothesis on some decline. However, there is, as Tara mentioned, there is no evidence to support these in terms of data, unfortunately. And so, we are driving this analysis, and Tara is driving this analysis based on data available. While some of the claims are interesting, there are other things that could be leading to these change in sizes. And I want to reiterate that what Tara has found is that there is a lot of variability. It's not like a single river and all the rivers showing the same pattern. No, what we are seeing is, based on the data that we have, we're seeing very different patterns. If you look specifically at a given river, you see a lot of variability. How much of this is driven by the data, the way it's collected, the observation noise that we have? There is more information on that, and in the paper there will be some documentation of this. And I think we have to be careful with making these assumptions that right now what is driving the lengths is some supposedly unreported catches. It happens for sure, but there's probably more critical takes of fish in rivers right now, in specific rivers, than in the ocean, that are more critical, because these fish are coming back to rivers and are still being exploited. Be it in some places, some fish that are being caught and being held and then released, that generates some mortality. In the context of climate change, we know that this increases the chance of either worse fitness for the female or just survival. And also there are some exploitation that remains in certain population in New Brunswick, in Nova Scotia, and in Quebec. There are some harvests that exist, and in some places they take quite a bit of fish, and that also impacts the population. And this is probably more documented than what's happening at sea. So yeah, I think we have to be careful. Like the big drive, like what we can notice is that the exploitation rate has significantly dropped over the last 50 years due to the moratorium, and that's the key thing. This is documented. Yes, maybe there is still some poaching at sea that happens, but before like really putting all my my eggs in this basket, I'd love to see some supporting evidence, like some satellite imagery of ghost fishery gear that is there. Until then, I'm gonna leave this as more an opinion than and a theory than something that happens.
Okay, thanks, Guillaume. I think that's the end of the questions. So great presentation. Thank you so much, Tara, for taking the time today to join us and present your results in your presentation. As you all know, for those that been coming to sessions before, we do record the session, so it is available on our YouTube page at the end of today. So if you know someone that missed today and wants to come in and see and listen, send them to our YouTube page. I also want to take the opportunity to mention that we have one more webinar before the end of 2023. It's going to be on December 13th with Rachel Duval. She's going to come give a summary on what is called Wild Atlantic Salmon Strategy, and it's what we heard. So she's going to give a summary of that, of where they're at. So please come join us in December. So once again, thank you very much, Tara, and everybody else that came to join us today. So thank you. So see you soon.